the COVID vaccines. I'm going to start off this discussion by telling you how the body's genetic code works. Genetic means inherited code. This code is inherited from our fathers and our mothers. Half of it comes from the father, half of it comes from the mother. It has code in it for every single substance that is necessary for our survival. This code is carried on genes. The genes all linked together make up our DNA, and the genes code for individual substances that we might need from time to time. A message is sent to the nucleus of the cell saying that a specific substance is necessary. It then stimulates the specific gene that will create that substance to produce mRNA. Now that gene contains code, as the DNA contains code. The DNA contains all the code. The specific gene contains the code that is necessary for the substance that needs to be produced. That portion of the DNA then creates what is known as messenger RNA. Now, messenger RNA is different from DNA and can never be integrated into a DNA. For that matter, the only place where RNA can be transcribed into DNA is where a retrotranscriptase is present, such as with the HIV virus, because the HIV virus's RNA can produce a retrotranscriptase. This retrotranscriptase then produces DNA that eventually can produce the virus again, and this DNA is then inserted into the genome. This cannot happen in the case where mRNA is produced. The mRNA is the messenger. So the gene, the code that is in the DNA, sends a message to the ribosome. The ribosome is the factory, the place of production of what the body needs. And at this place of production, at the ribosome, the mRNA attaches, and it attaches at one end, and then it is actually moved across the ribosome so that the ribosome reads the code and then creates what the code wants from the body. This code then can produce all sorts of things. In the case of immunity, when you look at the B cells, at those cells that are responsible for the immunity that creates antibody, it will create a protein. And this protein that is known as an immunoglobulin. The immunoglobulin is an antibody, and there are different types of antibody, and these antibodies then attack whatever this has been formed against, and in many cases can kill it and can prevent infection from occurring. Now, these specific building blocks are all used by the different vaccines. In the first place, there is a live attenuated vaccine. Live attenuated means that the vaccine is a living virus. It is the virus that causes disease, but the virus has been weakened so that it will not cause the full-blown disease, and because it cannot cause the full-blown disease, is safe for the body to have it enter. Under rare circumstances, this virus can mutate to become a virus that causes the full-blown disease, and this happened in the case of the polio virus. So this tends to be a somewhat less acceptable vaccine than the other forms of vaccine. The second form of vaccine that is available is that of a killed virus. These viruses that are killed then also stimulate the immune system to produce antibodies and to produce an effect. These first two types of vaccine, the live attenuated vaccine and the killed vaccine, tend to be less effective than the other more modern vaccines, and for this reason these less effective vaccines need the addition of a so-called adjuvant, a stimulator, something that stimulates the immune system to produce a more effective immunity than would occur if the adjuvant were not there. The second type of vaccine, in other words, not the live attenuated vaccine and not the killed vaccine, which is the whole virus that is used, but the so-called vector vaccine takes another virus, a virus that causes very much less severe disease, and it builds into it the genome for the product that can create immunity. So in the case of the coronavirus, this product is the spike protein. And what this then does is the adenovirus is then multiplied greatly outside the body with the gene for the spike protein inserted into it. When this great amount of virus has been generated, the virus is inactivated so that it cannot cause disease, and then the cells in which the viruses were grown are filtered away. These cells are aborted fetal cells. These aborted fetal cells come from the 1970s, and the reason for the abortion is not that well known. These products are then put into the body, and these products then go to the cell where they generate the whole cycle and eventually lead to the formation of the spike protein. The spike protein is pushed out of the cell, lands in circulation, and the B cells attach to it, and the B cells then create antibody, as we have already described, and these spike proteins also attach to so-called T cells. The T cells create what is known as cell-mediated immunity. So then you have two types of immunity that come about. The first is humoral, circulating antibody that we've already described how that comes about, and this can then kill the virus in the specific way by actually causing a cascade of different substances in the body to attack the virus. And it also creates the cellular immunity, and the cellular immunity uses the cells to kill the virus. We will not go into the detail of all of this. The next way in which the immunity can be produced is by giving the body the messenger RNA, giving it the messenger that it will eventually go to the ribosomes to produce in the ribosomes the spike protein. And the spike protein is then extruded from the cell, and exactly the same happens as happened before. Now, the live attenuated vaccine, there is a Chinese live attenuated vaccine that is not available in South Africa at this stage. And then there are the vector vaccines. Those vaccines that are created in another virus, and this virus is known as an adenovirus, inserted into which is the genome or the message that creates the spike protein. There are two vaccines that work in this way. The first is the Johnson & Johnson vaccine that was produced by Janssen in Belgium, and they are a filial of the Johnson & Johnson company. And the second is AstraZeneca. AstraZeneca is the so-called Oxford vaccine, was developed at Oxford University, and they therefore lead to the production of mRNA from DNA, and then the mRNA goes to the ribosome, and at the ribosome it produces the spike protein, and the spike protein causes immunity. The other type of vaccine that one gets is the so-called mRNA vaccine, and of the mRNA vaccines there are two. The first is the so-called Mrna vaccine, and the second is the Pfizer vaccine. So that these four vaccines that I have just described all lead to the production of spike protein and then stimulate immunity in that way. The mRNA that is given to patients cannot be transcribed back into DNA and therefore cannot cause mutation. Now, mutation means that the genetic code is changed to cause disease. That cannot happen because mRNA has no way of being built into DNA, and the mRNA is removed from circulation within 2 days. Now, these vaccines, obviously, as all medicine, have side effects. And the side effects that we're looking at, in the first place, the side effect that comes from the injection is at the local site. There can be redness and pain, and there can be a local inflammatory reaction, a local allergic reaction, and then there can be a systemic allergic reaction. The systemic allergic reaction is known as anaphylaxis, and anaphylaxis can lead to death because it's a sudden fall of blood pressure, a crash of the circulation system, and it can end in death. For this reason, all patients who get the vaccine are to stay at the vaccine station for half an hour, so that if this happens, they can be resuscitated and no damage will be caused to them. Obviously, death does occur in patients when one is vaccinating very large portions of the community. Heart attacks occur, clots to the lung occur, irregular heartbeat occurs, and these can all cause sudden death, but they are not related to the vaccine as such. Particularly the two vaccines that I mentioned first that create spike protein directly from the genetic mechanism with the so-called vector vaccine with a virus that is unrelated. These two vaccines are known for the fact that they can cause coagulation problems. They can cause clotting. Now, the first case of which this clotting was reported was in Scandinavia, and it was in the large venous sinuses, in the large veins inside the brain, and obviously that was not that well accepted by the medical community. But other clots can also occur. Therefore, patients who have a tendency to clot should be protected against this and can be protected against this by the administration of anticoagulation, of anti-clotting substances. What also can happen is that one can get a flu-like disease, and then the heart muscle can be affected, but the incidence of these things is incredibly low, so that the chance that one can get damage from the vaccine is much less than damage from the infection itself. And therefore, the vaccine is preferable. The natural infection also is named for the fact that clotting occurs. Now, this clotting that occurs in less than 0.1% of patients. That is less than 1 out of 1,000 patients. It is not quite clear what this means, whether the vaccine itself really stimulates that much coagulation or whether it is a red herring. It is said that the spike protein crosses the blood-brain barrier, and when it crosses the blood-brain barrier, that it leads to mutation in the brain cells, and that these brain cell mutations then lead to dementia and other central nervous system problems, and that people die within 2 years. There is no proof that this happens at all, and to be quite frank, proteins don't do this. There is no protein that is known to cross the blood-brain barrier and lead to mutation and then lead to an excess of death. This is the information that I want to present to you in connection with these vaccines, and I hope that it will help you to make a logical decision about the use of these vaccines for yourself.